Hello, my name is Gustavo Garay. I'm with the University of Puerto Rico Mayagüez campus. I'm with Kara Dean and Dr. Jade Mitchell from the Michigan State University. And I'll be talking about quantitative microbial risk assessment with the dose response models in inoculum concentration of microplants interaction. So talking about a little bit about the big problems. Climate change has changed obviously the environment, but that makes the growers more concerned about the risks of the exposures of pathogens in their crops. Since then, we have to develop new tools to the exposures of these pathogens within the interactions of plants and microbes interactions. To help with this, we need to use the platform of QMRA. The QMRA framework is a risk assessment tool that estimates human, animal, or in this case, plant exposure to pathogens via water, air, and other environment. These have been recognized by the World Health Organization and by the agency, the Environmental Protection Agency from the United States. One of the main components of the QMRA is the dose response model. That is a quantitative mathematical relationship between the exposure to dose and the risk infection of the pathogen. Even though it was studied before, we haven't seen, for the best of our knowledge, any example of this recording or of this from this risk assessment. And by that, we're going to use a paper to study the two dose response models equation that will help us because they're the most biological possible one. And then we'll apply them to the dose response model classically using the QRM to plant pathogen interaction data. As you can see on table 2, we have two equations from the dose response model. These were chosen because they are the most biological possible one, making our main objective to apply this for to model plant pathogen interaction. And if they can be applied, we can combine datasets for the pathogen species and the host type, able to see if they can be pooled. To complete this, we chose an article from Smith, who has been working with three pathogens from the genre Phytium, and he showed us with the result of the mortalities with the crop with different concentration of each species, giving us the correct format to work in this study. Smith used 8 crops, each of them with 3 different species of the genre Phytium, and tested them for mortality in different concentrations. He did the same with 6 variants of tomatoes, both with one species. What we did is use his result graphs and with the web plot digitizers to create the format that we wanted, just like it seems on the figure 1 and on table 1 as the example. We had a total of 31 datasets, which each dataset we run it in the software RStudio, in which we use a code previously used for those response models test. This software will give us the result of if the models were a good fit model and in which of the model will be the best fitting model. It will give us the minimized dose to infect the crop and the acquired dose to infect 50% of the crop, in which what we call the M50. As you can see in the result, for the good fit model and the best fit model, beta poison seems to be the most optimal dose response model to be fit. In the case of the tomato variants, NIG in the majority seems to be in the lack of fit, but with a few of them having the beta poison as the best fitting model. For the result of our second objective, in which first we combine all the datasets with hosts and pathogens, it resulted that all of them can be combined, telling us that the 3 species can have the same mechanisms of infection. Even though these models are applicable, beta poisons seems to be the most good fit to the majority of the plant pathogen data. In the case of tomato variant, we know that maybe the lack of data for higher doses could be the most problematic issue to be fit. And even though the unrepresented the most susceptible response, searching for the literature, we know that these pathogens could be the most highest pathogenic for the onions, and they're so. Onion is a very susceptible crop for these pathogens. We need to continue searching for more data with the format of those responses to continue doing more tests for different pathogens. And for the future, we need to advocate researchers to do more tests in this type of format so we can continue to develop this type of tool. I'd like to give you special thanks to this PhD student Cara Dean for helping me so much, to Dr. Jade Mitchell for letting me in this opportunity, to Bianca Davila Montero, Matthews Moreno, and Alexi Chuizano for helping me in this shrub internship. Thank you.